Hello everyone and welcome to week number 10. So this is actually one of my favorite weeks as we get into a topic that I'm actually extremely excited about, and it's a subject that I am very passionate about, and that is phlebotomy, blood collection, and capillary punctures. So for this week you have two chapters that we're looking at, chapter 44 and chapter 47. And I'm actually going to go in reverse order again. I'm going to talk about chapter 47 first, and then I'll go into chapter 44. So chapter 47 is talking about phlebotomy and blood collection. And you may hear me at some points say phlebotomy, or you may hear me say venipuncture, but please don't be worried, don't get confused. They're the exact same term. They mean the exact same thing. It's just depending on what comes out of my mouth first, either phlebotomy or venipuncture. But phlebotomy is the collection of blood through a tiny incision in the vein, and this is typically done with a hollow needle. So let's start off by discussing what your role is as a medical assistant during this procedure. So during phlebotomy, you will be properly collecting, labeling, and possibly either processing or storing a specimen for processing. Now, in order to do this, you need to have some basic understanding of the circulatory system, the equipment that we use during phlebotomy, the topic of asepsis, and the venipuncture procedure. Now, quite a few weeks ago, probably in weeks 2 and 3, we talked about asepsis, both medical and surgical, so I will not be covering those topics during the video. If you want to renew your knowledge on those, please make sure that you reread this portion of the chapter along with the rest of the chapter. So let's start off by talking about the circulatory system, and this is actually a very important system to understand because it's talking about the heart, blood vessels, and blood itself. Your heart has one main job, and that is to pump and keep your blood moving. Your blood vessels, on the other hand, their job is to provide a one-way travel route around our body and then back to our heart. There are some key players, though, with the blood vessels that we need to talk about. Those are arteries, capillaries, and veins. Arteries are what take the blood and away from the heart. So their whole job is to take blood away from the heart, and arteries typically have thick, elastic walls that feel very hard on the outside. When they carry blood, it is bright red in color, and this is because there's no oxygenation in that blood. Arteries do have a pulse, so when you're trying to distinguish what the difference is, make sure you understand that if it's got a pulse, that's an artery. Next, we have our capillaries, and they act as sort of the gas station for your blood. They fuel, or they give oxygen to the arterial blood and into the cells, while the garbage, or the carbon dioxide, is removed from your cells and carried into the veins. Once in the veins, they carry the blood back to the heart, but first they will be delivered to the lungs, where they kind of drop off that carbon dioxide that we breathe out, and they get that new supply of oxygen breathed in. Veins have very thin walls, and they feel very spongy, almost like kind of a bouncy ball, or similar to a trampoline. The blood found in your veins is kind of a darker red, and that's because of the oxygenation that it has picked up. So now that we understand the blood systems, let's talk about the phlebotomy equipment. So there's a wide variety of equipment used for phlebotomy, depending on what type and what procedure you're going with. You can use things such as blood collection tubes, double-ended needles, tourniquets, tube holders, and syringes. Now, your blood collection tubes are also called evacuated blood tubes, and they're designed to automatically withdraw the exact amount of blood needed for testing. Each tube is color coordinated according to the anticoagulant or additive found within it. These tubes can be either glass or plastic, but plastic is more widely used because it's safer not only for the medical assistant, but also for the patient. Double-ended needles are needles that have a tip on one end that will puncture the tube and the other end that will puncture the patient. The tip of the needle is called a bevel, which is where the cut or a slant is. Remember, when you're drawing blood, you want the bevel to be facing up towards the sky in order to properly collect a sample. The lumen is the space inside of the needle, and most commonly when you're drawing blood, you're going to be using a needle that is 20 to 21 gauges in diameter. Tourniquets are elastic bands used to help collect a sample. They will be placed about 2 to 3 inches above the puncture site and should only stay on for about 1 minute at a time. Any longer and it starts to have inaccuracy into the sample, causing problems that will make your sample incorrect. Tube holders are a plastic device placed at the end of a double-sided needle to hold the needle in place and hold the tube in place. Syringes come in all sorts of shapes and sizes, and it depends on what size sample you need in order from what size you will go from. Typically with blood draws, you're going to use anyone that's 5 to 20 milliliters. So now that we've talked about the procedure, we've talked about the circulatory system, let's talk about the actual procedure itself. So the procedure can be performed in several areas of the body, but most commonly you're going to use a space that's called the antecubital space, or the bend in your elbow. There are 3 main veins that we use for this procedure: your median cubital, your cephalic, and your basilic. The median cubital is the most popular because it is the largest and typically found right in the middle of that antecubital space. The cephalic is the second most common. It's not nearly as large as the median cubital, but it is better anchored than that of your basilic. You can find the cephalic vein on the outer edge or the thumb side of your antecubital space. Last is that basilic vein, and this is the least commonly used because it is not well anchored, but also because it is right above your brachial artery. If you go too deep, you can nick that artery. This vein is located on the inside edge or the pinky side of your antecubital space. So, now that we know where the procedure takes place, let's discuss the 3 different types of venipuncture procedures. First one we have is your vacuum tube method. This is also called the evacuated tube method. This is the most common method because it allows multiple samples to be obtained at the same time without multiple sticks. The vacuum in the tube is set to the exact sample needed for the amount of blood per procedure. Once it's filled to its max capacity, it will stop filling. Next, we have our butterfly method. This is also called a winged infusion method. It is a needle that attaches to tubing. At the end of the tubing is where a syringe or a tube holder would go for evacuated tube systems. This is typically used for smaller veins that are more difficult to draw from, either on children or cancer patients or elderly patients. And lastly, we have the syringe method. Now this method is used to collect samples from small pharyngeal veins that cannot stand the pressure of the evacuated tube method. The drawback to this method is that the amount that can be taken is only limited to the size of the syringe that you do have. When you're drawing any sort of method, it's extremely important to make sure you follow the order of draw. The reason is to prevent cross-contamination between tubes with the skin, bacteria, and other additives. As you can see here on the screen, this is the correct order of draw right here. The other one that can be replaced for blood culture tubes here at the top is an actual yellow top tube, and then everything else would be the same. Do not confuse yellow with this guy here. This is actually considered gold. So the last thing I want to discuss in this chapter is capillary punctures. This is also called a skin puncture, and it is used when a very small amount of blood is needed. The appropriate sites for adults are the middle or ring finger, third or fourth digit, on either hand, but preferably your non-dominant. When you use these fingers, you want to use the outside fatty edges. You can also use the earlobe or the infant's heel, but typically after the age of about 1 or 2, we stop using the heel for infants. There are 2 types of collection devices for capillary punctures. We can use capillary tubes or microtenor tubes. Capillary tubes are designed to pull the blood into the opening of the tube. They can be glass or plastic. Typically ones that have a blue rim are those that have no additive, and red means that they have heparin in them. The microtenor tubes are essentially just smaller versions of the evacuated tubes that we use for larger blood draws. Alright, so let's talk about Chapter 44. So Chapter 44 is talking about clinical labs, and for this it's important that you know why the clinical lab is so important. It's helpful not only to the physician but also to the patient by providing a way to screen for diseases, to confirm conditions suspected by the physician, to rule out a condition, establish a baseline, monitor the effectiveness of medication, and to assess the progress of a disease. Laboratories generally fall into one of 2 categories, either a qualitative or a quantitative. Qualitative test means that it will either give you a positive or negative result, for example, a pregnancy test, where a quantitative will give you a numerical value, for example, a glucose test. It's also important for us to discuss and understand some of the important lab regulations. The first and most important one is that of CLIA, the Clinical Laboratory Improvement Act of 1988. It was created to protect patients by creating safe policies and procedures. The amendment addresses the issues of quality control, quality assurance, record keeping, and qualifications of personnel performing testing. It establishes a standard for testing, accuracy, reliability, and timeliness. Another amendment that you need to know about is OSHA, but we have talked about this in great depth, so I'm not going to go too far into that. But we do need to discuss quality assurance and quality control. Quality assurance, as a medical assistant, you are really responsible for carrying out the procedures that involve planned and systematic activities to ensure that the requirements are met and results are accurate. Quality assurance will include quality control, orientation, documentation of controls, maintenance checks, required calibrations, temperature checks, checking expiration dates, and proficiency testing. Quality control or quality assurance is also developed what we call reference values or normal values. Quality control is part of the quality assurance, and it focuses on the physical proof that results are accurate. This is done routinely by performing mock testing on either calibrations or control solutions. Calibrations are specifically prepared test strips or cartridges that are designed to produce a predetermined result, where quality control solutions are chemicals that produce an expected result and are similar to a testing specimen. Alright, so that is the wrap up for week 10. As we talk about your homework this week, I want to talk about your discussion post first. That will be due no later than Tuesday by June 6th at midnight. And what you want to do is explain the importance of the order of draw, what happens when it's not properly followed, and do your blood collection tubes need to be filled completely full when drawing blood? If yes or no, wherever you fall in that, you need to give your explanation as to why. Please make sure that you do reply to at least one classmate no later than June 10th, which is Saturday by midnight. And then for your quiz this week, you do have a quiz over the information this week on phlebotomy and blood drawing. It's a 20-question multiple choice or true-false quiz, and you have a 20-minute time limit for that. If you have any questions or concerns, guys, please make sure you're reaching out to me either course messages in Blackboard or my direct line on the Rockford campus. Have a great week 10, everybody. Bye-bye.